Good afternoon. My name is Michael Dickens, and I'm a research scientist with HPRC. And I'm going to go over an introduction to Galaxy, and we'll go through a short analysis, and I'll introduce some of the features that we have. So these are some of the topics we'll cover. I'll start out by, if you're not familiar with usegalaxy.org, this is the group that created Galaxy. It's a large community. There's lots of users that will contribute to the project. They'll contribute in the form of creating tools. They also contribute in the form of creating online tutorials, both tutorials that you can follow along in a web page and also videos. We'll go through the process of uploading and downloading data and managing histories. I'll show you how to switch tool versions, and then we'll look at the file attributes. And then one important thing is how to permanently delete files. Permanently deleting a file will remove it from the file system. That way, any non-essential file is not taking up space on the file system. You can delete a file and then you can undelete it. What you'll want to do is delete the file, and if you're sure you don't need it, or if you've waited a period of time, then you don't need it, then you'll want to permanently delete it to remove it from the file system. I will show you a little bit about job debugging, and then what happens when you copy data between histories. And then the last part will be hands-on running, creating a variant calling analysis. And then I'll show you, introduce the workflow, how to create one from your history and then create a new one. All right, you can go to usegalaxy.org. This is the main website. This is hosted by the Galaxy team. So this is actually an instance of Galaxy. It's run on their computers. You can browse, you can get a free account, you can browse tools, you can run some analysis and save. If you want to save it a long term, you'll need to create an account. Otherwise, you'll have like a temporary history. So you can go through here and see how Galaxy works and test it out to see if it's a good fit for your project. Like I said, there's a lot of users, a big community. They share workflows, they share tools. There's a lot of help, a lot of tutorials and videos on their webpage. We can actually access their tutorials from within our Galaxy instance. So it's all web-based, so you use your browser and upload and download and view all your work on their servers. Here's the web page that has their tutorials. A lot of these are follow along in text files and on the web page, and there's also some videos that go along with that. So you can browse that, and there's a lot more tutorials than we could ever cover. So you can get a lot of information from there. Okay, we have our own Galaxy installation. It's on Grace. It's called Maroon Galaxy. And first, you'll need to get an HPRC account. Once you get your HPRC account, you can request to be added to Maroon Galaxy. If you're off campus, you do need to use the TAMU VPN. And then to log into Maroon Galaxy, it's the same login as you log into HPRC, your NetID and password. We also have a user guide that will cover some of the concepts and also some things you'll need to remember when running some of these tools. Also, remember there's no backup of Galaxy files. So if you want to back them up, you'll want to download the data and store it locally on your desktop. All right, so this is the main web interface. So what we'll do is go to, once you get your Galaxy account, you can go to our main page, and there is a user guide for Galaxy here. So just go to the Galaxy user guide. And we've recently updated to version 24.2. And you can get a link to Maroon Galaxy right here. So This is the interface. You can see we have some best practices. Read over this at least once so you'll get an idea of quotas and FTP uploads. There's also a link to the docs and slides here. There's some other links here. Basically, these are links to shared training and tutorials. A couple of known issues here. You can read through those. Try to avoid parentheses in the names of your files. That can cause issues with some of these tools. But also, you also want to take a look here because when we have a maintenance, we'll have an orange box like this posted that their maintenance is coming up. So jobs may not start if they're scheduled for like 7 days and the maintenance starts the next day. Okay, this little graduation hat, you can click on this graduation hat and it'll take you to the Galaxy training site. And from within here, from within this page, you can actually access the any of these tutorials. Let's just look at microbiome. Here's one for 16S nanopore. Just click this and you can see as an overview, and then it has information, it has an agenda, asks you questions. You can click this open to see the solution. And then you can click on the outside of that box and it will you'll get back to your main Galaxy page. All right, just went over our knowledge base. Let me go back over that. So I want to go over here within Galaxy. You can see here we've got information on downloading files, uploading files, deleting files. So you can read through that if you need to. All right. One thing you'll want to do is check your HPRC SU balance. We have a utility for that. You'll just go to the Galaxy and then click tools. If you're not here already, if you click it twice, it disappears. So click HPRC category and then click HPRC SU balance. And then we just want to show the balance. This is a quick script. It really doesn't take 24 hours. You can actually drop it down to 1 hour. And you can actually drop it down. There we go. And then we'll just run that. So notice I have an unnamed history. By default, you'll have an unnamed history. You can change this name. But we'll just go ahead and run this tool. Now, when you run that tool, you'll see it. Still connecting. You'll see it show up over here in the right panel. This is a history of all the tools you've run. It starts with item number 1, and I'll show you how many items you have. So you can also show your balance, or you can show the stats for all your jobs. What that does is it shows you all the jobs you've run this fiscal year, the time, duration, the job IDs, and the service units charged. Okay. So now it started, there's one job added to the queue. It doesn't show up here. Maybe you can hit refresh here. Let's just hit do a refresh of the web page. And you can also change your account, your default account. If you have 2 accounts and you're running low on one service units on one account, you can switch to the other. All right. While that's loading, switching software versions. Whenever you open a tool, it defaults to the newest version. If for some reason you need to rerun an analysis and you need to use the older version that you ran the first time, you can click these 3 boxes here and select the version. Otherwise, you can just accept the newest one. Okay, so that job is queued and waiting to run. So let's look at, if you go to tools. Let's just search for SAM tools. So we'll just look at the first one, and then actually there's not more than one version. Let me try maybe BW has more than one version. Yeah, BW has more than one version. So you can click here and you can see the versions here. For some reason you want to use an older version, you just select that and it'll tell you it switched to the older version. Otherwise, you may just want to keep the latest version. All right, we talked about the graduation hat to get to the tutorials. We also have disk quotas. Everyone starts out with 1 terabyte of disk quota. And this is displayed up here in the top right, what percent of the 1 terabyte you've used. Click on that, it'll show you some more details. You may want to hit refresh at least once. It might retotal, it may show a lower value. You can also get some information here, but usually it's just easiest to go into your history and delete what you don't need. All right, there's also favorites. You can add tools to your favorites. The only thing I found out is that some of these tools, if you add it to your favorites, you're unable to uncheck this star, so it'll remain in your favorites. So just choose your favorites wisely. All right, so some of the features Galaxy has is that you're able to select the number of cores in memory and the amount of time for each job, kind of like if you're running Slurm jobs on the command line. The thing about some of these tools is some of these tools only support single core processing. So if that's the case, actually, let me find one that SAM tool stats that only supports single core. If it supports single core, and that one doesn't. No. Probably these collections here. Okay, if it only supports a single core, you won't have an option to select multiple cores. You only have an option to increase the memory and increase the time. Now, if you see in the documentation that it does support multiple cores, but you're unable to select it here, please let us know, because we have to actually enable the multi-core processing for software that supports multi-core. So we install a tool, but then we have to verify that it supports multi-core before we enable the multi-core option. And when you see the multi-core option, you're able to select either the lowest 1 core 7 gigs of memory or the entire compute node, which is 48 cores and 360 gigabytes of memory. Otherwise, single core processing, you can only select memory and time. We also have a few tools that may require more than 360 gigabytes of memory. So we have some large memory nodes on Grace. In that case, that tool can be configured to use up to the 2.93 terabytes of memory in all 80 cores. We don't allow this option on all of the tools, because this large memory partition is reserved for jobs that require a lot of memory. Mainly, this is going to be genome assembly tools that need a lot of memory. All right, let's just say you want to run a tool and you've seen it online, but we don't have it on Maroon Galaxy. You can go to Toolshed. This is the usegalaxy.org Toolshed, and you can search for tools here. Some of the tools in this Toolshed are not on usegalaxy.org, and they're not on our Maroon Galaxy, but they are in the Toolshed. If it's a recent tool, some tools may be old. It we may have some difficulties running these tools in Galaxy, but we can still install and test and try it out. But for the most part, if you see something in here, or if you see some tool online in an article, a research article. You can go search here to see if this tool is available for Galaxy. If it is, let us know and we can install it for you. All right, let's talk about uploading files into Galaxy. Let's go ahead and see this job is completed. So I'm actually able to see my balance. And then I'm going to go back to our main page here. All right. So you can rename a history here. Just click the pencil, edit, and really not my first one, but just showing you how to change that. All right. So now you'll be able to identify what the history is all about. All right. So uploading files less than 2 gigabytes, you can do that through Galaxy's web interface. Let's see. Okay, so there's a few different ways. When you do upload, you're going to get this page here. So there's a few things you can do. You can choose a local file, and this is going to take you to local, meaning that's your desktop that you're working on, not Grace. So you're going to upload a file from your desktop or laptop. Choose a remote file. This is actually linked to your FTP directory on Grace. So these files have to already have been uploaded to Grace using an FTP client, and we'll show you that in a little bit. And then another one you can do is paste fetch data. Now this one you can either paste the URL of a file. For example, maybe you have a list of chromosomes or a list of maybe a genome assembly, and it's UCSC and fasta.gz. So you can type in that whole URL or copy and paste the URL in there, and then click start, and it will upload it for you. The format for the file that gets uploaded, Galaxy will auto detect what type of format it is. It will know whether it's a compressed file or uncompressed file, a text file, a tab delimited file. So that's a feature, a newer feature that we have now, as opposed to our first installation of Galaxy. All right. So I'm just going to work on this slide to show you about copying a file between histories. Now, when you actually copy a file, let's just say I'm in my unnamed history on the left here, and from this Canoe 2 file, history, I can click this file, left click and hold it and drag it over here into this history. Now, when I drag it into this history, it's not making a copy of the file. So you're not doubling your disk space for that file. But it does show you that history has 212 megabytes of data. But your overall history doesn't increase, your overall usage doesn't increase. So Galaxy has some built-in functions that doesn't basically just make copy of files every time you copy it to a new history. It just keeps track of where the original file is located. So that can be useful, so you don't have to upload your genome assembly into each history that you're working on. Okay, so for files greater than 2 gigabytes, we recommend that you upload using FTP client. The one that works best for Galaxy is this Bitvice. You can install that in your Windows computer. If you have a Mac or Linux computer, you can actually use an SFTP command in your terminal. But for Windows, I recommend Bitvice. If you have some files on Grace already in your Scratch directory and you want to copy it you want to import it into Galaxy, you can use the SFTP command on Grace and copy it to your FTP directory for Galaxy. And there's some information and documentation on how to do that on our user guide, Galaxy user guide. So what happens is after I have uploaded a file using SFTP or Bitvice, it's in my FTP directory on Maroon Galaxy. Then when I click upload, I can click choose remote file, and then I'll be able to see that file right there. So then I select the file and then I click start, and it will move the file into my history, and it will remove it from the FTP directory. Okay, this is the one I mentioned earlier where you can just paste the URL into this box. Again, upload, then go to paste fetch data, paste the URL. You can actually do more than one URLs, one per line, and then click start, and it will add those to your history. All right, so let's do let's run an example of how to import data into your history. So we're going to use the SRA, NCBI SRA sequence read archive utility, and we are going to import an accession for sequence reads that are on the NCBI server. So what we're going to do is go back to log into Galaxy. So do you have a Galaxy account? Do you have an HPRC account?
Yes.
Okay, I think I added you to this morning to Galaxy. So let me if you want to just go to our main website, and then on the there's a user guide to the left here. Galaxy, and then there's a link to Maroon Galaxy. See if you you're able to log in. Okay, so in this exercise, this will download FASTQ files, read pairs 1 and read pair 2, two files. It'll also create a log file for an accession. Now, sometimes when data is in SRA, it comes as paired files. It might be single in files. This will handle the file formats for you. So go ahead and search for faster in the tools. Search faster. There's a couple of SRA tools, but this one actually is a little better because it'll format the files better than the original tool. So faster, and then you'll see a tool that says faster download and extract reads and FASTQ, and then select that one, and in the field accession, see there's a star by it. I mean, it's required. So in accession, go ahead and type in the SRR 3175-4396. Once you have that accession, you can scroll to the bottom of that tool and then click run. And it should only take a few minutes. Good. Okay, so let's look at the data. The first one's a log file. If you click on that, it'll expand here to the right. If you click this little eye, you can actually take a peek at the data. There's only 3 lines here. If it's a really large file, like 100 megabytes, it'll only show you the first chunk of data. So it tells you how many reads were written. You close that one. And then other data, there's nothing here in this one. And then single in, says there's 0 data sets because this is paired in data. There are no single in reads. Paired in, there's actually a list with a pair. If you click on that and then click on that, you can see the forward and reverse reads. You can take a peek at the data with the eye here. It shows you it's only showing you the first megabyte. And then you can go back to the history up here at the top. Now, another thing you can do is you can click this Windows Manager here. If you click this Window Manager, it'll stay on. It'll stay yellow clicked. Then if you view one of these files, let's go to back here. And if you click View, it pops up in a little window that might be easier to manage. If you view the other one, you have multiple windows. So if you need to compare a couple of files, this is a good way to put them side by side. Not much you can do with FASTQ files other than look at the format. But if you have some other output files, like some tab files or some summary files, that's a way you can pop 2 different files out and compare them visually. All right, I'm going to go back here and then back to our main page. Okay, so let's talk about deleting non-essential files. So the first one is a log file, kind of really don't need that one. Second one is some other data. There's no other data, and the single in that's empty too. So click on the little trash cans for each of those. So the log file, other data. We're going to click. We'll click collection only. Single in, collection only. So we're left with the paired in, and then our balanced one. Okay. So now we've got these deleted. They're not showing up here, but if you click this garbage can up here, it says, show me what files are deleted. So if I wanted to, I could undelete them if I did not want them deleted. I could put them back into my history. And if I click this, then it goes back to my main history as not deleted. I want to delete it and remove it, and then go back and include deleted. You can see those two are deleted. Those three are deleted now. Now those are deleted, meaning they're not in your history, but they're still on hard drive taking up space. So if you want to purge is like permanently delete. So purge all deleted contents. You click the little gear. Gear is right here. Purge all deleted contents. Of course, this is delete. You can't get it back. That's okay. We'll click OK. All right. Include deleted. All right, so that's a way to delete, permanently delete, remove from disk if you don't need it anymore. So SRA data, when you download SRA data and you start to move it and you do processing, say you did an alignment of the data, you don't need the original files, right? Because you can always they're stored on NCBI servers. Let them pay for the storage costs. Go ahead and delete the files you downloaded since you've already processed them, right? Okay. It's taking so long. Okay. So now we're left with this paired in data. We're going to look at FastQC. FastQC will give you some analysis of the quality. So in the tools, we want to search for FastQC. FastQC, very first one, FastQC, read quality reports. Now it says, please provide a value for this option. Raw read data from your current history. Well, there's no compatible option because this is actually a list. So what we need to do, we need to click this folder icon here. Now it shows me the list. Okay, we're going to run this list. So it should run on both files. So run the list, and then you can select, go to the bottom. You can leave these as they are. If you want, you can move this to 1 because FastQC is really quick. It's only a summary, and then click Run Tool. FastQC will take a summary of the first like 200,000 sequences. So if you have a really big file or you have a small file, it runs about the same because by evaluating the first 200,000 sequences, you can get an idea of the read quality. So let that run. See what's next. Okay. So when it is complete, you're able to see some reports like this. So this doesn't change the data at all. It's just evaluating. It doesn't do any trimming or anything. It just looks at the quality. And anything you download from the web, you definitely want to run this tool on that data because you don't know where the data came from or the quality of the data. You need to make sure it's good. So I'm going to just look at the tool FastQC. Something I mentioned here is at the bottom. There's a lot of useful information about the tool, the outputs, and there's actually a tutorial here on how to run this. There's 42 tutorials available which use this tool. You can see how popular this utility is, this tool. All right, on this one, you can re-enable this Windows Manager. Otherwise, it will just show up in the middle here. So I'll just go ahead and look at this one here until that's finished. So one thing about an older Galaxy version is that you had to convert files to FASTQ Sanger because there are two types of FASTQ files. The older sequencing platform was not in FASTQ Sanger, so you had to convert it to FASTQ Sanger. There's actually a tool that did that for you. You had to go and click and convert it, make the conversion. But now Galaxy will automatically determine whether it's in the newer format or it's in an older format. If you download something from SRA, it might be in the older format. But Galaxy handles that for you. But you can see now it's in Sanger 1.9 format. There's the number of sequences, 5693. The sequence length, so this is 36 to 151. GC content. So over here are the summaries. The green, of course, means good. Yellow, caution. Red means there's something that's not expected there. So you should review that. And we'll look at that. Okay, so let's look at we don't need to look at the web. Let's look at the web page for forward. Okay, so here is the quality. You can see the scales from 0 to 34. And this is base 1, this is base 151. So as expected, the very first part of each of the sequence reads has very good quality, and then towards the end, the quality starts to go down. Whether or not you need to trim this or not depends on what your downstream analysis is. Some tools like GTK recommend you don't trim them, and some other tools you may trim to get better quality. But there's a couple of failures here, and one is this right here, pair-based sequence content. So if you notice, there's this, you should expect to see G and C as complementary. So you should expect to see GC content about the same, and then the AT content about the same. But right here, there's this weird pattern here. So just from previous experience, I can tell that this is a sequence motif for the way this DNA was prepped. It's a transposase insertion site, so it recognizes a certain motif, and that's why you see this at the beginning of this read. So it didn't really fail, but you just need to be aware that what's going on here. And if you're not sure, you can look more into how this sequence reads were prepped in the lab. So this is okay. It's part of the DNA. It's part of your DNA that you sequenced. It's not a contaminant. And so that's also what the sequence duplication level is too. All right, so that's that. Let's go back to the beginning here. So we're going to delete this history. It's something you can easily reproduce. So the three bars here, we're going to delete this history. Okay. Yes, I want to permanently delete the history. Click OK. This actually brings me back to a different history I created, but I'm going to create and start a new history here. All right, so y'all deleted that one? Okay, you can always go back and make it history again. Okay, so some files that you upload can be in this FASTQ Sanger format. You click the pencil icon so you can see the format. If the format is FASTQ Sanger GZ, now some tools will accept gzip format, some tools will not. So if you need to unzip the format, you click the pencil, you edit attributes, click on data types, and then select FASTQ Sanger, convert compressed to uncompressed, and create the data set. And then it will create a data set that's in FASTQ Sanger, not FASTQ Sanger.gz. So what you want to do, since now you've got the original compressed, and you have the decompressed files, you want to go back and delete the compressed files. Okay, because you can always take the uncompressed files and you can compress them later, or you can just delete them if it's something you downloaded from SRA. So the way to do that, similar way, you click the trash can and then go and purge all deleted data. So just be aware of how you're managing data in your account, so you don't have compressed and uncompressed of the same data. You can delete one of those sets. All right, history divider. This is just a simple tool that will create a line in between histories. So sometimes you run a utility and it creates 5 output files, and you run a different one, it creates another maybe 6 output files, and they're all stacked on each other. So if it's hard to tell where one tool stopped and the next one started, you can run this and it will just draw a line between jobs. And we'll do that in a little bit. Okay, shared libraries. This is something that we can add. I don't believe users are able to add shared libraries, but if you ask, let's say you have a data set you want a group to work with, instead of having everyone upload the same data set, we can upload it as a data set, and then we can allow only your group to access that data. But we do have some global accessible data for everyone. So you can go to libraries, and then you can see we have a genomes, and we have a number of genomes here. Now, some of these genomes are already built in to some of the tools, and you can select them within a tool, but you may need another file. So then you can import files into your history. Now, remember, when you similar to when you copied a file, if you import any of these files into your history, it's not actually using up disk space. It's just creating a link to the original file. All right, failed jobs. It happens at times. Here's one where it says could not queue job. Most of the time it's because you're out of service units. So if you see this, just run your my project command and just reduce it to one hour. That way you don't get charged. Even if you've used up all your service units, then you won't even be able to run the my project command. Then you can just send us an email and we'll let you know. All right, here's another one. Sometimes you don't schedule enough time for the job. Maybe you scheduled 2 hours and it's right in the middle at 2 hours and it stops. You'll need to restart the job with just go ahead and make it 12 hours. Give enough time for the processing to complete. This one ran out of memory. Just increase the memory and try again. If you let us know, well, it'll tell you it ran out of memory, and then if you click this, it looks almost like a recycle sign. Click it and it'll run the tool again, or it'll load the tool again with all the parameters that you selected before. Then you can go to the bottom and increase the memory. That way you don't have to reselect all your files. So click that those 2 circular arrows and then increase the memory. Different ways to view errors. Click on I think the bug is different now. So click on the eye. I should highlight the eye. Click on the eye. It'll bring up this panel in the middle, and then you can expand some of these. There's these little arrows, and you expand them, and it'll give you it'll allow you to look in the standard out and standard error files for error messages. All right, sometimes this has happened with the other Galaxy. I haven't seen it yet, but if the little disk icon is missing and you can't download data, just refresh the browser. This one I've noticed with the newer Galaxy, if this reload button is red, just go up and refresh your browser at the top. All right, lots of videos again on doing a lot of different things. A lot of time and effort was put into that. So check out the videos. Maybe for some analysis, somebody may have already done it in Galaxy, and they may have actually a workflow, which you can import a workflow and it's already set up. So you don't have to go and build it yourself. All right, now the last part, what we're going to do is we're going to run through an analysis, and so you can get an idea of how what you can do in Galaxy. So I want to create a new history named, see, this is Candida dublinensis. This is a fungal genome. So create a new history. I've already got a new history. I'm going to call it, if you don't have if you're in a history, you can create this plus button and it will create a new history. See, I just gotta check the spelling. Dublini. All right. So I have a new history. All right. So import sequence reads from libraries. Have it here. Okay. So let's go to libraries again and go to libraries in the left panel here. Go to genomes. Here's the Canada. It's a diploid fungal genome. Click that one. Now you've got 4 files here. You've got features. This is a GTF file. This shows you the gene coordinates. You've got the genome assembly FASTA file, and then you have some sequence reads. So click this box to select all of these 4, and then we're going to add to history. And we're going to add them as data sets. Okay, and then select the history. It probably has a current history. You can go ahead and keep it at that and click import. All right, so now we have the data to work with. What I'm going to do is go back to Galaxy, go to Tools and HPRC. Galaxy Tools, HPRC History Divider. And then click Run. That will just give you a little marker to know where the original files start. All right, that's really quick. So does everyone have this set up like this? All right, next step. We're going to trim adapters. So search in Tools for Cut Adapt. Now, one thing about Galaxy is whenever you click and you're running a tool, if it's gray or if it's yellow, you can still go to the next step. It will know that there's something running, it's expecting some output. So what do we have? This was paired in. So click paired in. Paired in files. Now the other one we had was a list. These are individual sequences here. So you can see file number 1. It just populated what it found. So let's change that to R1. And then FASTQ number 2, make sure that's R2. So you need to have R1 first and R2. The order is important. Okay. So a lot of this, unless you have some special adapters to add, you can insert adapters. Otherwise, it has a list of adapters that it knows about. So you can scroll the bottom and then click Run Tool. All right, so that's great. We can go on to the next step. Align to a reference genome. So we can search tools for BWA. So let's start a new search for BWA. Now these reads, we didn't run FastQC, but these reads are going to be greater than 100 base pairs. So click BWA mem. Okay, when you align reads to a genome, the alignment tool needs an index of the genome, so it knows what to expect, how many chromosomes are in there. So we don't have a built-in genome index, so we're going to use a genome from the history and build an index. So it automatically finds a FASTA file. If you have multiple FASTA files in there, make sure you have the correct one selected. So let's have that. Should be number 2 for the second file we imported. And then just let BWA decide the best algorithm to use, and then come down here. The paired files, notice it has the same number 7 here. So we need to make sure we select read 1, which is number 6, and read 2, which is number 7. Even though those haven't completed yet, we're able to select them. So we can go ahead and keep building our analysis without, we don't have to wait for it to complete. So let's come down here. And we're going to use 24 cores and 180 gigabytes. This will just speed it up a little bit. If you have a larger genome, you'll want to use the whole node, which is 48 cores, 360. But this is a small genome, so we can get by with this, and it will complete within a few minutes. So we'll click Run Tool. All right. So while that's processing, we can go to the next step. So SAMTools flag stat, there are some flags within alignment files. It will give you some information about whether the 2 paired files are on the same chromosome and the correct orientation, and they're not too far apart. Okay, and it'll also tell you whether or not it aligned to the reference. You'll get an alignment percentage like 98%. If you see an alignment percentage of 5%, that means you probably have sequenced one organism and you're trying to align it to a different organism, and there's just not a lot of trees that are aligning. In that case, you'll want to check what type of, you'll have to find a way to see what, you can blast some of these sequences to see what organism pops up. All right, so SAMTools flag stat. So let's come here and search SAMTools flag stat. I think SAMTools stat might be the same, but we're going to do flag stat, the actual command. So right now it finds a BAM file, even though it's not run yet. It expects it to be there. So we'll just leave it at 8, number 8, which is the BWAMM BAM format, and then keep it at 7 gigs in 1 hour, and we'll run that or 24 hours. All right, when that's complete, we'll review that. But what we want to do now is we want to run Freebase. Freebase will call variants. It's really good on small genomes, bacterial genomes, fungal genomes. For larger genomes, like the human genome and mouse, GATK is specifically designed for larger genomes like that. But Freebase is a simple tool to use. On the command line, of course, is simple in Galaxy. So let's search the tools for Freebase. There's only one option here. So Freebase. And then choose the source for the reference genome. Reference genome, it's gonna be in our history. And then the BAM file, that's gonna be number 8, which will, when it's created, then this Freebase will run. And use the data set, that's going to be the FASTA file, the genome assembly file. Do we want to limit anything? You can go through all these parameters and decide if you want to set any limits. So Freebase, let's go ahead and make it 48 cores, and it'll still get done very quickly. So select 48 cores 360. We have the reference, and then click Run. All right, the next step is snpEff. snpEff is a tool that will annotate variants. So the Freebase is calling the variants. Since we have a reference genome, we know what the DNA sequence of the organism is, right? And if there's been a lot of research on it and we know where the gene coordinates are, then we can actually see if a variant, which means a single nucleotide variant, or maybe a frame shift or a deletion insertion, if that has any effect in a gene region. If it's in a intragenic region, like between genes, it might not have as much an effect, but we know that there are enhancers that may be in that area. But for the most part, if it's inside of a gene in a highly conserved coding region, then chances are it might disrupt the protein. So what snpEff does is it will annotate and let you know if the variant changes the amino acid sequence, or if the amino acid sequence is not affected. So that's snpEff. So we need to create a snpEff database first. So let's search snpEff. And then snpEff build. Okay, for some reason it's in here twice, but it's the same tool. So you can see we're going to use the latest version, and then name for the database. Canada, the actual genome build, the reference build is called CD36. So I'll put that in the name. It's down here, CD36. All right, input annotations, we have a GTF file. When we select GTF, it finds the GTF file for us. So we need to know, this is just a name, but we need to know the name of the reference. We don't have a locally cached one, but we can get one from the history. So if you select history, it finds our genome assembly FASTA file. Genetic coding, select genetic code for the sequence, we'll keep standard. And then that's it. Click Run Tool. So this sets up the database for us to actually run snpEff. So notice this one started before Freebase started. snpEff doesn't depend on Freebase to create the database, so it's able to launch right away. All right, annotate the variants. The variants, they are in the Freebase. Remember, Freebase called the variants. Now we want to annotate it. This one, we still can look for snpEff, and then we want to do snpEff EFFF. Okay, annotate variants. This is going to be the data sets 2 and 7, Freebase. I have it number 10. It's number 9 on the slides here, but we created a history divider, so it's number 10 here. So now number 10 just started, but we're able to select it. Input format. We're going to leave it at VCF. Genome source, locally installed. We don't have a locally installed one, but we do have a custom database in our history. And that's the CDUB CD36 that we created. So that should be selected. Okay. And everything else stays the same. At least for this example, you want to read through all this and figure out what all this other options are in case you need them for your annotation. Or yeah. And then I think snpEff only used a single core, so we go ahead and click Run. Okay, so then once this is done, we'll view the results. Just take a few minutes. Everyone's got this running snpEff. Okay. This is the history item. How many items you have in this history? So the first thing we have is 1. You'll probably have maybe 12 or 13. That's you probably had some other stuff. That's fine. We have some users that have 100, 200 items in their history. And if you delete them, it might go from 1, 2, 3 to 72, 73 because they deleted a bunch. That number will stay the same. That's helpful if you have any questions or if you need help, if you just let us know the history name and the history item number. Like give us the history name and then number 12, for example, because you might have several failed tools in there. We need to know which one you're asking about. So history and the number, I can find it quickly. All right. Yeah. If you change the history or no, it will not. They're separate histories. Even if you copy data over, you can delete this history and the Galaxy knows that to keep the file because it's in a different history. Okay, that's done. So let's go ahead and enable this window manager again if you haven't, if it's not enabled already. And then let's click on HTML stats. This one. You click the I and you can open that and it will show you, here's your genome name, the date it was run, the snpEff version. This is the actual command that was used. And then there are some warnings. Usually that means maybe can't remember exactly what the warnings were, but usually those are something like the variant falls within a certain region or another. But those aren't failures, those are just warnings. But you can see the average variant averaging one variant every 263 bases. Shows you each chromosome 1 through 7, and then there's R. It shows you how many variants and the variant rate. Total SNPs, MNPs, insertions, deletions, and some other ones. There's no inversions or duplications here. All right, it even shows you the type of variant. How many? There are 865 variants that have a stop gained. This means there was a variant that changed the codon from some amino acid to a stop codon, which could affect the protein, especially if it's in the beginning of the gene. Synonymous variant, a lot of those, but usually that's not going to affect the protein structure, right? Because it's the same amino acid. It's just a different coding for the amino acid. All right, so that is snpEff analysis. Now, let's just say you have several samples that you want to run the same analysis. So you can actually extract this as a workflow. You don't have to go, you can do two workflows 2 different ways. You can either run the analysis to make sure it works and then extract the workflow, or you can go and build the workflow, save it, and then run the analysis, make sure it works good, edit the workflow as needed. So what we can do here is click the 3 bars and extract the workflow. So you can, it gives it kind of a long name. I'm going to change this to variant calling snpEff, because I'm gonna use this on a different organism. Okay. It shows you what history items will be included. So we want all of these steps to be in the new workflow. So create the workflow. So it already created it. So you can edit or run the workflow. So let's go to over here on the left, you'll see workflows. Workflows, these are your workflows. Here's one I created 4 days ago. It's the same. I went through the steps to make sure it works. Here's the one I created a minute ago. So now if you click, let's see, click run the workflow. No, we don't want to run it. Click. I think you just click the workflow. Yeah. If you click the workflow, and it will show you a preview here, and you can see the input files run into cut adapt, and you have these little lines here that connect the output of one tool into the input of another tool. And since we've already defined this when we work through it, it already has the lines drawn for you. But if you're creating a workflow from scratch, you'll have to drag those little lines, and I'll show you that in a little bit. All right. Well, that's loading. You can share this workflow with others in your lab, find workflows, import them into your account, and then you can run the workflows. All right, so here it is. You can actually see it's already kind of locked in here. This is just a preview. You can click edit if you need to change anything. You can see the way they have it. It's a nice flow. If you move any of these around, those little noodle connectors can get look like some roads, so they keep them tucked in here, so they kind of where they go. So we're not going to edit this one, so let's go back to workflows. Let's exit here. Let's see. There's probably a cancel here. Maybe there's a cancel edit. Nope. If you do make any changes, make sure you save it here. So we're not going to make changes. We're going to exit leave page. All right, so permanently delete this history. You can do this the same way. Delete this history and permanently delete the history. If you want to keep it up and review it for a little bit, you can do that. You know, you can always go through and redo it. If you want to view some of these output files before you delete it, that'd be fine. Even here, even though these are the ones we imported, we don't need them because we have the cut adapt files. That is actually using our disk space because that's a new file. The ones I just deleted, those were imported, so those aren't using up disk space. So we could actually delete them, and it's not going to change our disk usage, but it's just be aware of how that works. So anything you copy or you import from libraries into history, that's that usage is from wherever you got it from. Okay, any questions? Okay, the last slide is if you want to create a workflow. So let's go to workflows and then the top right, create. So workflows, create. So there's first thing we'll do is give it a useful name, descriptive name. Maybe you can make it generic so it's not locked into a certain sample or genome. That's taking a while. Fail to workflow. Let me start over here. Refresh the page. Well, let's take a like a 10-minute break. We're almost done, but looks like we're having some network slowdowns here. Let's go ahead and take a 10-minute break and then come back. Oh, is it loading? Okay, so let's see if it works now. Workflow, create. Yeah, okay. We'll skip the break because we're almost done here. So workflow name, my second workflow. Now we'll give it something more script. We'll call it sequence read QC. We'll use uppercase QC. And okay, we'll leave it at that. And then you could give it an annotation. We'll do QC and say you want to create a workflow that always runs QC and cut adapt. So when you start a new history, you can run your workflow and then you can continue to work on other items after your workflow is complete. All right, so now let's go to inputs. Oh, we'll go to tools. Let's go to tools and then we're going to search FastQC. FastQC, click it once, it adds the tool over here, and then you can move it around in this canvas space here. Notice it says unsaved changes. So click save there in case there's a network issue. You don't want to lose your 1 hour of work. I don't know if it has an autosave feature. It might. So let's see. The next thing we want to do is cut adapt. Cut adapt. All right. So now this cut adapt, there's only, let's see, if you come over here, it shows you what options. So let's just say you're going to make it paired in. So now it expects 2 files, one from a file number 1 and number 2. Okay, so FastQC. See what I have here. Oh, okay, cut adapt. So you're actually going to have kind of like a branch here. You're going to have the input. Let's just say input data set. It's going to be data set 1. There's another one, data set 2. So this is going to be data set 1. I wonder if you can set the format. So we'll go ahead and put this data set 1. Draw reads from your current history. Okay. So we're gonna look at FastQC for just one of these. But we don't need to feed FastQC into cut adapt. We just we can build it like this. So now it's going to run 2 different. It's going to run, at least for the first read, it's going to run FastQC so we can view it. But it's also going to be input into cut adapt. And then also for each of these tools, make sure you're not missing out on anything that you need. It's a way to build it here. You can even set the runtime parameters. So cut adapt doesn't need a lot, so we can leave it at 1 core and 24 gigs. Then BWA, so we can search for BWA, go back to tools, BWA mem, we'll select that once. And then so our original files get trimmed, and then we're going to have BWA. It's going to, let's see, paired in reads. So this is the first set, it's a second set. So this output from cut adapt is going to go into BWA. And here's your output file, okay? And that's just a simple workflow. We'll save that. And then if we go back to our main page, go to our workflows, you can see there's our sequence read QC workflow. Now we can start a new history and then run the workflow. Okay, so we don't really have any input files yet. You'll need to have input file first. What we could do is go to libraries, genomes, and import these two. Add to history as data sets. Unnamed history. Let me go ahead and rename this so we'll make sure we're getting it into the right one. This is a test workflow. So let's do add to history, test workflow import. Now if we go to our workflows, on workflow, just make sure you select read 1 for input 1, read 2 for input 2. And then you can run the workflow. All right. So that can save you a lot of time if you have a lot of sequences to analyze. All right, so that's pretty much it. If you have any questions, I'll be here for a while. You can let's go back to workflows. Let's edit this workflow. I think if you hold control, you can zoom out a little bit. You can make a really big workflow. You don't have to hold the control. Just use the mouse scroll. What is it?
I do let me know the percentage, so I can make workflow like this, and it's like I can use it for a minute.
Yeah. Yeah, just as long as your sequences are relatively the same as far as the amount of memory. Sometimes some input sequences are not so big and others are larger, and it might use up more than 7 gigs of memory.
I mean, if there's an error with it, I mean the error, like we know like where it's from and why, like when you're running it.
Yeah, so what it'll show you is for example, if number 5 fails, if cut adapt, let's say if FASTQ fails, then cut adapt will not run because it doesn't have the output files. So FASTQ item number 3 and 4 will be red, and the others would probably be gray or maybe blue because it's paused. It'll let you know which one failed. Yeah. If you actually go to the portal, go to jobs, active jobs. You can see FastQC and CutAdapt. FastQC just completed, CutAdapt is running. So it's using Slurm job scheduler in the background. So even though CutAdapt is completed here, oh, it's still running. FastQC is completed. CutAdapt is running. Yeah, FastQC. All right. Another useful thing is history multi-view here. You can see you can click this panel and slide it over. So it only shows about 4. It only shows 4, but you can add a new one here. Create and pin a new history. You have to select which history. Oh, select history. So you can select one of your histories to show them side by side here. This is where you would copy data sets. So if I wanted to copy this to here, I can just click and drag. Oh, we'll copy this with it. See how it copies? All right. Right now the current history is unnamed, so I'm going to switch back to my test workflow. All right, that one's got the one more step to go. Oh, something went wrong here. Oh, what we forgot to do is index. I didn't create an index from a history. All right, so I need to edit my workflow. Remember the one step we had was index. Select the FASTA file to index. I didn't do that. Let's see what happens. Oh, I didn't select this. I should have selected. But I don't have it here, but I can import it from the libraries. Yeah, so workflow, go back to your workflow and edit. You'll need to add one more data set. This one's going to be let's see, this one needs to go here. So now, here it is. Use the following data set as a reference genome. Here's this. This is what we need right here. I have so many lines. It should just be one. That's weird. Okay, save that. Now, you'll have to have the genome FASTA file and then select it here. I can actually just import it. The test workflow. Now I can rerun this tool and then click the FASTA file. Just make sure you have read 1 and 2, cut it down 1 and 2. Yeah, because when I click run job again, it keeps your configuration like that. Actually, probably should have clicked more cores. All right, that's it. We'll hang out a little bit in case you have anything else, but that's it for the session. Yeah, usegalaxy.org. They have that's hosted by Use Galaxy team on their own computers. Maroon Galaxy is on our computer that we manage. It's on campus. Yeah. And also, sometimes if you get onto Use Galaxy, since it's public for everyone to use, everyone in the US, maybe even other countries, it can get very busy at times, and it might take a while for just to launch jobs. Yeah, you get disk spaces last here. You can start at 1 terabyte, and we can increase the space if you have a bigger project. Okay, thanks for coming in. Here's our information. If you have any questions, you can send an email. It generates a ticket in our help desk, and also there's a link to our short course survey